400 million. I mean, just take a second to let that massive number really sink in. We're talking about the astonishing number of liters of blood that the two human ventricles combined are going to pump over an average 75-year lifetime. To kind of put that into perspective for you, that's enough fluid to completely fill a whole lake. A lake that's a kilometer long, 40 meters wide, and 10 meters deep. It's just a staggering volume to even wrap your head around.
So, welcome to today's Explainer. Today, we're totally shifting our perspective. We aren't going to look at the cardiovascular system as just, dry, memorizable biology. Instead, we're exploring it as a literal masterpiece of biological engineering. I mean, it's this incredibly sophisticated, self-repairing system of pumps, pipes, and highly specialized fluid that literally keeps us alive.
Okay, let's dive into this. Here's our roadmap for today. We'll start with why we actually need circulation, then we'll get into the physics of blood flow, how we measure the machine, the actual makeup of blood, and finally, we'll wrap up with the viscosity paradox.
Right, moving into our first section, why we need circulation and the evolutionary problem of size.
So, you might be wondering, why do we even possess this complex plumbing system in the first place? Well, it all comes down to the evolutionary consequence of body size. Think about an isolated, single-celled organism. It doesn't need a circulatory system at all. It survives perfectly fine on just simple diffusion, exchanging nutrients and waste right there with its external environment. But us, as complex mammals, we've got a massive scale problem. The distances separating our central cells from the outside world are just way too long for diffusion to work. A simple tube simply wouldn't cut it. The nutrients would be entirely depleted by the time they reached our core. So we evolved this rapid convection network, and it's driven by a dual pump. The right side of the heart acts as a boost pump, sending blood to the lungs, and the left heart acts as the main pump, driving it all the way out to the rest of the body. This maintains a really steep concentration gradient, which makes absolutely sure our deepest cells get oxygen and clear waste instantly.
Section 2. The physics of blood flow. We're talking pressure, pipes, and pose wall.
Now, blood flow in our vessels is normally smooth, silent, and energetically efficient. This is a state called laminar flow, where fluids move in these nice, undisturbed concentric layers. But there's a tipping point, and this brings us to the Reynolds number. It's a dimensionless mathematical quantity that dictates exactly when smooth blood flow suddenly tips over and becomes chaotic and noisy. When your Reynolds number is below roughly 2,000, your blood flow is silent and perfectly organized. But the second it exceeds 3,000, boom, those layers break down into wasteful, noisy, turbulent vortices.
So what actually triggers this? What pushes our blood past that tipping point into turbulence? Well, there are a few specific culprits here. First, a large vessel radius, like what we see in the aorta, our largest artery. Second, high blood velocity. This happens when your cardiac output spikes during, say, heavy exercise. And third, low blood viscosity, basically thinner blood, which can happen in conditions like anemia. In any of these scenarios, the disruptive inertial forces completely overcome the cohesive forces holding the fluid together, and turbulence just erupts.
The absolutely crucial takeaway here is how wildly sensitive this entire system is to the actual size of the pipes. And this brings us to Poiseuille's law. It's an essential concept in hydrodynamics. According to this law, viscous resistance to flow isn't determined by the material of the vessel wall, but entirely by the fluid's friction and the vessel's geometry. And here is the mind-blowing part. Flow is proportional to the fourth power of the vessel's radius. The fourth power. That means even a microscopic tightening of a blood vessel causes a massive exponential drop in the amount of blood flowing through it. It's seriously an incredibly precise control mechanism.
Moving right along to Section 3, measuring the machine and how doctors listen to the system.
You know, whenever you get your blood pressure taken, right? The doctor inflates a cuff to intentionally stop your blood flow, and then they slowly release it. Well, what they're listening for with their stethoscope are called the Corot-Cough sounds. They are literally listening for the physics of turbulence we just talked about. In phase 1, as blood just begins to squeeze past that tight cuff, it creates a sharp tapping sound, and that marks your systolic pressure. Then in phases 2 and 3, it becomes more of a swishing murmur, and then a louder thumping as the turbulent flow continues. In phase 4, the sound kind of muffles, and finally in phase 5, complete silence returns. That silence is key. It means the vessel is fully open again, the Reynolds number has dropped back down, and laminar flow is completely restored. And that marks your diastolic pressure.
And this brilliantly illustrates another way we measure the machine. Doppler echocardiography. Just like how police use radar to bounce sound waves off a car to calculate its speed, doctors can actually bounce ultrasound waves off moving red blood cells. Notice how the red color here encodes blood flowing toward the transducer, kind of like blood moving through the mitral valve during diastole. Meanwhile, the blue color encodes blood flowing away, like blood moving toward the aortic valve during systole. It's literally a real-time velocity map of your heart in action. Pretty amazing, right?
Okay, Section 4, the makeup of blood, a highly complex fluid.
Let's shift focus from the pipes and the pump to the actual stuff flowing through them. If you take a sample of blood and spin it in a centrifuge, it separates into these distinct layers, revealing what we call the hematocrit. The hematocrit is basically the fraction of the total column occupied by red blood cells. That's typically around 45% in an adult male and 40% in an adult female. Sitting right on top of those red cells is a really thin, whitish-gray layer called the buffy coat. That makes up about 1% of the blood and contains your white blood cells and platelets. And then, the top 55% is this pale white, watery solution of electrolytes and proteins known as plasma.
But the absolute stars of this fluid are for sure the red blood cells, or erythrocytes. Just look at the unique biological engineering of their shape. They're biconcave discs, meaning they're indented on both sides. And this isn't some random accident. This specific shape gives them a much larger surface-to-volume ratio than a simple sphere ever could. It absolutely maximizes the diffusion area and minimizes the distance gases actually have to travel inside the cell. Plus, to make room for a massive concentration of oxygen-carrying hemoglobin, mature red blood cells literally eject their nucleus and mitochondria entirely. They are tiny, perfectly optimized gas transport vehicles.
Finally, Section 5, the viscosity paradox and the mayonnaise metaphor.
All right, let me ask you a highly provocative question. When you think of a thick liquid flowing, you probably picture something like honey, right? But what if I told you that blood is actually way more like mayonnaise than honey? Why is that?
Well, it all comes down to a property called yield shear stress. See, water, saline, and honey are what we call Newtonian fluids. If you dip a spoon into a jar of honey and pull it out, gravity alone is enough to cause the honey to drip in a continuous stream. It always flows. But if you dip a spoon into a jar of mayonnaise, it just sits there in a big glob. It effectively has infinite viscosity at low forces. It requires a specific, hard push, a threshold force, to break its internal structure and start moving. Normal, whole blood is non-Newtonian. It behaves exactly like mayonnaise. If the force pushing it is too low, your blood just won't flow. It clumps together and remains completely immobile.
So what's the actual culprit behind this bizarre non-Newtonian clumping behavior? The answer lies in the interaction between your normal red blood cells and a specific plasma protein called fibrinogen. Now, fibrinogen is absolutely essential for blood clotting, but its simultaneous presence alongside your red blood cells creates this exact yield shear stress we're talking about. Without fibrinogen, your blood would flow just like a simple Newtonian fluid. But with it, the cells cluster, increasing their effective density and creating a fluid that requires a serious, heavy push from your heart just to get moving.
To wrap this all up, our survival relies on this incredibly precarious, perfectly engineered balance. You've got a constant pressure generator in your dual pump heart, you have a dynamic, highly responsive set of containers in your blood vessels, and you have this smart, non-Newtonian liquid organ, the blood itself, that knows exactly how to clot, flow, and deliver oxygen right where it's needed.
So, I'm going to leave you with this. Knowing the intense physics, the massive evolutionary pressure, and the sheer mechanical force required to pump 400 million liters of non-Newtonian fluid through thousands of miles of microscopic vessels, how much will you appreciate your very next heartbeat? Thanks for diving into this explainer with me, and I'll catch you next time.